So hello everyone, welcome to this series where we discuss one UPSC Prelims PYQ at a time and also talk a little bit about the topic so that we are future ready for any question from this particular topic. Now, the question that we are going to discuss today is this one, but before solving it, let us first understand what DNA barcoding is and why do we need this particular technology. In nature, there are a number of species that look very alike to each other, making it very difficult for us to distinguish between them. But unless a species is properly identified, its usage in understanding evolutionary relationships, developing biodiversity conservation strategies, and crop improvement strategies would not be possible. To solve this problem, the scientists came up with a technique which is called DNA barcoding. The technique uses one or more short standardized regions in the DNA of a particular species, which is highly conserved within that particular species, but show variation with other species to generate a barcode. Just like the product barcode that we find in a store or the fingerprint of a human, the DNA barcode is a unique identifier of that particular species. So let us understand how this particular technology works. First, a small sample of the tissue of the species which is to be identified is taken. Then the DNA is extracted from the tissue. Then the specific target region is amplified using PCR technique. The amplified region is then sequenced. So sequencing basically means reading the nucleotide sequence present in the amplified region. Then the nucleotide sequence is compared to a reference library of DNA barcodes. If it matches with the reference barcode of a particular species, then the species is identified. So what can be its applications? So applications could be species identification, biodiversity research, conservation strategy development, and also to check the safety levels of food by identifying microbial contamination or adulteration. Now, let's come to this question. So the question says, DNA barcoding can be a tool to A. Assess the age of a plant or an animal. No, this is not right, because can our fingerprints help us in understanding our age? No, it cannot. Can a barcode tell us about the expiry date of a particular product? No, it cannot. So yes, it cannot assess the age of a plant or animal. Distinguish among species that look alike? Yes, that is the very idea of DNA barcoding, right? The third is identify undesirable animal and plant products in processed food. As we just discussed, it is also used to understand the safety levels by identifying microbial contamination. So this statement is correct. So this brings us to the answer, that is 2 and 3. Thank you.